What is synthetic microbiology and why is systems biology central to the development of this exciting scientific discipline? Welcome to Editors in Conversation. This episode is brought to you by mSystems Journal, available at msystems.asm.org. I'm your host, mSystems Editor-in-Chief, Jack Gilbert. This podcast is supported by the American Society for Microbiology, which publishes mSystems. Make sure to press the subscribe button wherever you are listening or watching so that you never miss an upcoming episode. mSystems publishes the best research in single-cell and community microbial systems biology, and new computational laboratory approaches, and synthetic biology. For example, a recent paper, a high-throughput method for screening for genes controlling bacterial conjugation of antibiotic resistance, which presents a novel experimental framework to screen for conjugation-based horizontal transmission of antibiotic resistance between over 60,000 pairs of cell populations in parallel, enabling rapid screening of compounds that decrease transmission of antibiotic resistance. This can be found at msystems.asm.org. I'm joined today by Professor Pam Silver and Professor Danielle Tullman-Ercek. Professor Silver is currently Professor of Systems Biology at Harvard Medical School. She is also a core member of the Weiss Institute of Biologically Inspired Engineering at Harvard University. She has made significant contributions to cell, systems, and synthetic biology. Professor Silver has several major research efforts around bacterial engineering, for example, for ARPA-E. Her group focuses on designing biological systems for both health and sustainability. Recent innovations include bacteria that can sense and respond to their environment, and the bionic leaf. Professor Tullman-Ercek is an associate professor of chemical and biological engineering at the McCormick School of Engineering at Northwestern University. Professor Tullman-Ercek is interested in controlling the movement of materials across biological membranes with a goal of enabling and enhancing the production of pharmaceuticals, biofuels, and materials in microbes. Manipulating the transport of electrons, small molecules, and even signals across these cellular boundaries is a vital component of most synthetic biology applications, and she is developing the tools and methods to engineer membrane proteins and other protein superstructures to meet this grand challenge. So welcome, guys. I'm really excited. This is our first mSystems Editor in Conversation piece. Thank you for being here. So let me start. I'm going to start with you, Professor Silver. I'm going to call you Pam. Is that okay?
Yes, please.
Cool.
I never think of myself as a professor.
I always say professor makes me sound old. So I like to go with just Jack, but it works out better. So Pam, can you tell me what you think synthetic microbiology actually is?
Well, first, let me start by first celebrating the journal. I've been thrilled to be on the editorial board, and Jack, you're just doing an amazing job. This journal is really just doing everything right so far, and also wonderful to be here with you, Danielle. Great to see you again. It's been forever, right? So I want to go take this question back a little bit. Oh, and I also wanted to say a thank you to the journal for embracing synthetic biology or synthetic microbiology. That I think we've got to get more papers in this area.
Right.
I think it is one of the fastest growing areas in synthetic biology. But let me back it out for a moment and just remember what is synthetic biology to begin with. And everyone has a different view of that, but I'm going to go back like 20 years. I'm probably the oldest person on this call. When we originally, a group of us at MIT formed what we called the Synthetic Biology Working Group and articulated, I was the token biologist there. It was a bunch of engineers and computer scientists. And the question on the table was really, what's the deal? How can you make the engineering of biology faster, cheaper, and more predictable? And those words just resonated for me as sort of the defining components of synthetic biology. And we wrote a roadmap and this and that. And the nice thing is we actually achieved a lot of the goals of that roadmap, except I think the area that we really lag behind in, and I think is really about synthetic microbiology, is the predictability component. So I would like to see a time when we're getting deeper and more people are really striving not just towards making observations about microbia in consortia, of which there are tons of observations, but
Right.
how do we take that and synthesize it into a strategy where we can then build consortia with predictable properties? Building synthetic consortia or even single microbes with really what we predict what they're going to do. We don't just do it by chance. And we draw on the extensive knowledge we have of how bacteria behave. We know E. coli best. And so the other thing that I think we're going to see more of in this space is starting to engineer new organisms or new microbes. And there's so many of them out there. And so that's what I see happening in this space.
That's really cool. I appreciate that. Could you riff a little bit more on predictability? You're saying the ability to predict how changes you make in the microbes will influence their behavior, their molecular biological properties. Is that right?
Sure. So say you want bacteria. Well, let's take a recent paper that was published in mSystems by Ashley Shade, where she's really beginning to get at this question of what do you need to know to predict how, say, a 3-member consortia can function. So how can you have 3 bacteria that depend on each other to form a functional consortia? And believe it or not, this has not been done that successfully. You mentioned some of my DARPA work, and one of our DARPA projects was to make something like a 6 or 7 member consortia, which turned out to be impossible. So we've got to make DARPA, right? But we got to make a 3-member consortia with predictable properties, and then you want it to do use. So for me, a key element of synthetic biology is engineering things to do useful things. But sometimes that means making toy systems first. But I would like to see people really head towards useful things. And so I just want to go back to Ashley Shade's paper because she developed a beautiful system where so the bacteria in her system don't actually have to be growing next to each other. She can start to measure what they're secreting, the metabolites that they need to connect to each other, because she's separated the bacteria from what's outside them. And then this drove me to, I just became fascinated with her work. I hope she's listening. And I went to her website, and I think she really captured one of the reasons for building consortia. And she says, we want to understand how microbial communities respond to stress so that we can manage them quickly to recover. The capacity to recover quickly and fully from a large stress is called resilience. Understanding the rules of microbiome resilience will help support healthy outcomes for hosts and environments, for stabilizing essential functions, et cetera. So I think she really captured it on her website. So I don't
like
know
that.
Answer your question, but that's what I'm thinking.
Yeah, no, it does. That does. I think that's great. I mean, Danielle, if I may, what do you think is the connection there for? I mean, your lab is focused on really building an understanding of how the pieces that make up microbes and microbial communities come together to form I guess the molecular underpinnings of systems ecology and systems biology at the cellular level. How does that feed into synthetic biology? Is it just a toolbox, or what's the plan there?
Oh, I think it's a mutual feedback. So each discipline is informing the other, and that is how we're going to make progress. And so from my world as a synthetic microbiologist, when I'm trying to build something new, almost always we spend at least half of our time figuring out the regulation and all the inputs and environmental pieces that might be affecting the system that we are trying to engineer. And that's separate from the part, let's say we want to make an assembly at a certain time, do something new, but we have to make it at a certain time, right? And we have to be able to control that part. And so we're learning a lot from systems biology about exactly what regulation might feed in, what metabolites are important, what is going to be the input, and then we use that. At the same time, our systems can be then used to help inform what's going on within the system. As soon as we engineer it, we realize what else we don't know. And that in turn drives a lot of hypotheses to go back and look at that regulation in this example much more carefully. And the same thing that was for my work. We're still in the level of the lab. And so we're working on things on the single cell level, but I think it's even more complicated problem once you move into consortia. And that's where we really want all of these applications to be, I think. And synthetic microbiology, especially in the gut, is going to be really important. If we want to engineer an organism that is a probiotic organism in the gut, well, we have to get that to work in the lab, but we also have to get it to work in the context of all these other microbes. And so it's a really complex problem, and they feed one another. And so it's really exciting to work in it.
When you talk about feedback mechanisms, though, I mean, if you're dealing with that at the level of the single organism right now, right? Yeah, I totally understand, right? The idea of taking that organism, putting it into Pam struggling with a 6-member community, trying to model it in the gut would be next to impossible.
Yeah, how many? Well, we actually did a thing where we had 10. We did a mouse where we had, I think, 10 unique microbes in a gut where we tried to look at the impact actually of phage on individual components of that. It was purely an observational study, but it was somewhat defined.
Yeah, exactly.
And that was that was saying, do the phage knock out these bugs, and how does that control?
We were using phage are fascinating, by the way. So I think one of the things you wanted to talk about is more papers that mSystems can publish in this space, and I want to make a big plug for phage. I think phage research is going to have a reemergence. Because people are discovering phage all over the place. I'm really excited about all those phage in the ocean. What's going on there? All those phage.
Absolutely.
I want to see every one of those papers in mSystems. So what I love about phage is that they're a way to interrogate relatively specifically a microbe within a population. And so we tried to do that in the context of the gut. It was fascinating, the potential to knock out one member of a community. And then if that community is more defined, and ideally you could predict how it's going to respond, then that would really be a synthetic situation with potential therapeutic or other indications.
I like that. So let's tie that back in, Danielle, to the work that you're doing with the single cell organisms. So let's say we can develop phage and knock things out. The key aspect that I still see are these energetic feedbacks, the necessity to uptake nutrients and to pump things out which can be used by other organisms. And we talk a lot about that in ecology. What we don't really understand is how to quantify those feedback mechanisms. Can you talk a little bit about how you're trying to quantify feedback in order to better design these organisms and potentially communities in the future?
Yeah, I think there's a lot to figure out in terms of how to measure and quantify this. And that's probably the biggest challenge in the field, the very specific subfield that I'm speaking about now, right? This quantification, you can do it at the level of the systems. You can start now because we have this ability with data and machine learning, take in all of this data with as many observations as you can and use that. But the other way is a more traditional and I think just as valuable, and we should also have papers in mSystems on, is doing very detailed studies of exactly which molecules are taken up by which organisms and which processes, and finding ways to actually calculate that. It turns out it's very difficult to calculate how much comes in or goes out of any particular molecule or piece of information. But it's right there. We are so close to being able to do that at the point that we are now with all of the tools that we have in synthetic biology.
Well, give me a concrete example. How are we just at the cusp? What's the leading edge of that technology?
So the ability to engineer things like transcription factors and start to be very precise. For example, I'm going to give a different one, actually. So this is not awkward, but kind of fun to talk about. So Pam's my inspiration. She's the person always doing the work that I want to share. And one one effort that she was involved in was developing circuits to, and in this case, there was another sort of altruistic goal, but to develop biosensor triggers, figure out what promoters are activated by what states in the gut. And so this was work with David Rigler. And so synthetic gene circuits enable systems level biosensor trigger discovery at the host microbe interface, right? So this is an example of something where we can start to use our ability as synthetic biologists to construct circuits that we can then plug in thousands or even hundreds of thousands of different pieces and see what they do, what they respond to, and then also use that to interrogate the system and figure out within a consortia which ones of these signals are activated. So it's kind of going at both ways. But we have a lot of different circuits out there, and I think a lot more clever ideas to come using all sorts of different molecules and gene circuits like this.
Yeah, I think Danielle really captured, again, this goes back to our deep 50 or 60-year understanding of how bacteria regulate transcription, at least E. coli. And this is kind of the fruits of all of that research now, is this ability to engineer just about anything you want in E. coli. Now, that's E. coli, but we want to get there with other organisms. And I think that's going to be a huge challenge going forward. And I really want, even though it's hard and people always say, oh, work on the model system, it's the best, but we gotta move into other organisms, like reef organisms, for example. I'm really into the reef now. I think we need to save the reef and engineer bacteria of the reef. But I wanted to add another technological advance to what Danielle was speaking to regarding quantitation, is the realization and the ability to measure phenomena at the single cell level. And that, that's playing out in every aspect of biology, but it's also certainly true in bacteria. And, in consortia, is every member behaving the same? Or, what kinds of assumptions are we making that may not be true at the single cell level?
Yeah, but that opens up a can of worms, right? And I was thinking about that when you were saying, we've got E. coli and we can manipulate E. coli, but we still don't know what all the genes do in E. coli, and we still don't know how those genes respond in a single cell in a population of cells of E. coli. And so there are so many pieces that can add noise to that system. Are we dealing with an insurmountable problem?
Well, I personally don't think so, because I actually think, probably between Danielle and myself, if you asked us to design an E. coli to do something, we could probably do it. It probably wouldn't be perfect. See, that's the thing. Would it be perfect? Would it have a good on state, a good off state, et cetera? And that's where the predictions come in. But I'd be willing, give us a problem and let us go away, and we could probably figure it out. A transcription-based circuit, because come on, not to insult anyone, but transcription in bacteria is really well understood, and it's just wonderful. And that's why Danielle can do all these amazing things.
Yeah. And I want to make a plug for my other favorite organism, which is Salmonella. So E. coli is great.
Like E. coli, right?
But it is so much like E. coli. In some ways, it's like 99% just E. coli, and yet it has all sorts of other systems that we don't understand and regulation in place. And it just gives a taste of how much what we're learning in E. coli can be translated in some way to the other organisms, not directly sometimes, maybe by what tools we develop. But I think it's not insurmountable because we aren't starting from scratch now. Some of what we've learned, and it'll always be different things, will be portable into these other organisms and other contexts.
Well, this opens up a question I had about synthetic biology in general. You guys are doing amazing work, and there are lots of other people out there in the field doing this. Are you building libraries of organisms that have particular features that that people can use or access, so that people don't have to continually rebuild these phenotypes in future studies?
Am I
Well,
or
anyone
is
could answer
the field?
Boy, if my lab could keep a library of anything, I'd be. No, I mean, I was funny. I was just thinking I don't want to advertise, but we're trying to start a center here for synthetic biology. And in fact, again, moving away, so there's a thing, and you may have one at Northwestern, but there are these things called foundries where there's just that idea that you will have lots and lots of iterations. But I'm thinking, again, I want to go back to this idea of moving away from the physical setup to the predictable one, so that, again, if you're the naive engineer who wants to build X, instead it will all, in this dream world, it will all live inside a computer that came out of our heads and is now in a computer that can design that for you. And then you have all these DNA companies and you just order it and you reduce the number that you have to order. So in fact, I'll just use Danielle, mentioned our work on building an E. coli sensor for the gut. And it is based around my favorite phage, the lambda phage, which is really understood in detail and so well understood. So when we built this circuit, which can sense and then remember that the cells were exposed to a certain signal, and they were. And this circuit, I have to say, first time work. It's like works first time every time. I was a student in the Potashny lab when this was being figured out, and boy, did they figure it out. It's like the gift that keeps on giving. But people say to me, well, how many did you have to build? Did you build hundreds, 100? And I said, no, we built 5. and so that's an example of, if you really understand the system, and whether that's in your brain or in a computer, you won't have to build that many things. And so I want to get away from that mentality and more towards let's try to build as few and test as few as possible and have them be right.
Oh, I like that targeted approach. It makes a lot of sense.
Well, it's probably not going to happen for a while, except for lambda phage.
Right. Moving into mSystems, I know you mentioned a couple of papers, each of you. Do you have any other papers that mSystems has published that that you think are reflective of what we would like to see published in mSystems from a synthetic microbiology perspective? Maybe Danielle, go first.
Yeah, well, see, now this is another one of Pam's.
I'm seeing a trend here. I'm not sure.
No, to be fair, there aren't that many.
Yeah. Because we need more.
We just need more.
So this one's out of the Gerber lab. And that one is engineered interspecies amino acid cross-feeding increases population evenness in a synthetic bacterial consortium. So this is, again, returning to that idea of being able to control what's happening in consortia. And this paper is really interesting in that in nature, at least in all the observations we've been making of nature, of these natural interactions, we think about evolution as survival of the fittest, and you do the thing that costs you the least and you get the most out of it, but you might not survive then if something about the environment changes. So what makes you really robust? It tends to be antagonistic interactions, right? So what was really nice about this was it showed that when we're engineering systems, we can actually engineer better systems and better consortia, not just reliant on that antagonistic, but also beneficial interactions. And I think for me, what was really nice about this particular article was not even the science itself, which was really cool, but the idea that maybe we haven't seen all these other beneficial interactions because we just haven't been looking at the right aspects of the systems. And so I really hope to see more discovery of observations and then using those observations to build new consortia like this one based on amino acid cross-feeding. And I think there's a lot of opportunity for that with signaling molecules. I think there's opportunity for that with, let's say, one of my favorite cofactors, if you will, is cobalamin. So looking at all the coronoids. And I know there's a lot of work in those areas that would probably fit really well in mSystems as well.
I like that perspective, especially with vitamin Bs and trying to get them into the human intestinal system in a way that can be controlled and mediated. That could be huge. I'd really like to see that work. You mentioned, from my perspective, I thought that was particularly interesting, communication pathways, ways of managing quorum, for example, in microbial communities. And is that a major part of the synthetic microbial ecology aspects? Can you put bugs into a system to control swarming and motility of microbes in an environment?
Sure. Actually, one of the classic synthetic biology papers, very early work from Francis Arnold and Ron Weiss, I think. Correct me if I'm wrong, Danielle, was about just that, using quorum sensing actually to have bacteria make patterns on plates. Beautiful paper, years and years ago. Many of us, myself included, have built on that to try to use quorum sensing in a practical sense. For example, in the gut, can we engineer a bacteria that would sit in one place in the gut and then sense what's nearby, and when there's a critical concentration, then it would do something, for example. That turned out to be really hard. I wanted to go back to the paper you talked about that we did with Georg. That was so hard. That was the one where we tried to make like the 6-member consortia. And I just want to shout out for the first author of that paper, Marika Zizak. That was her PhD work. And boy, that work was so hard.
But you got 4. So
4. She was aiming for, that was the project where her PhD assignment, I think, was 6, and we gave up at 4.
So I want to say, as an undergraduate, over 20 years ago now, I had a project in a process control. I'm a chemical engineer by training, and it was process control, but it was a bioprocess control project where we had to keep 3 organisms alive in a codependent manner with the different feedback loops available to all of process control in the engineering world. And I think mine lasted the longest, but it was only a few weeks in computer time, but it was as a few weeks for 3 organisms. And so the fact that we're able to actually build systems now 20 years later that with 4 organisms and doing this in a designed way is amazing to me.
Barely. We have a long way to go. But to the point about your point about quantifying things that are needed, that really gets to the Shade paper that I mentioned earlier, because she really was getting to a system where in relatively high throughput, you could start to do just that and then make quantitative analyses of what's needed for cells to talk to each other or to grow together, and et cetera. So I think that really does follow nicely from what we were talking about.
I agree. And anytime I get to speak about Professor Shade's work, I'm excited because she is a phenomenon, a wonderful researcher. Pam, are there any other papers that we've published recently or in the last few years that excited you in mSystems from this side of the scientific domain?
Excite me. I don't know if they fit under this rubric. So I have to confess, I didn't spend a lot of time.
That's all right.
Hours in the day here.
Well, yeah, Danielle, do you have any other papers?
I'm too busy editing all those papers you send me, Jack. None of which, by the way, are in synthetic biology. So since when have I become an expert on gut microbiomes,
As an editor, we have to be experts in everything to the best of our abilities.
I guess I would just make the case that we can all make observations, but we could just start with a little more testing of the importance of those observations in a functional sense by reconstructing them. And I just really would like to urge people to start taking all these observations and trying to condense them into something where we start to use it. And I don't, I think I asked you this once before, but where are all these observations are they all being kept somewhere? Because, I mean, you must send me 3 papers a week on this topic. And I can't tell
one hopes that
where's it all being kept so that someday somebody can go in and really start to use it to make predictable engineering of cells. I'm not getting a sense of that, Jack. Maybe you could answer that.
Hey, I'm not under interrogation here. But yeah, I agree. The community does need to make sure that these data are publicly available and are available and accessible in a manner that can actually foment that kind of future research program. So
that gets to another issue that is relevant to engineering biology is standardization. So all that data, and I know I've pestered you about this before, but how's all that data going to be compared to each other and standardized so that the engineers, or are we just going to have to go back and repeat it all? I mean, what's the plan?
There is no plan. There is just science. But I think that's the perfect example, right? When sequencing became cheaper, it was easier just to resequence things. But when we talk about introducing new organisms into the toolbox, the play kit that we have for making synthetic organisms or synthetic communities, with so many of those organisms hard to breed in the lab and also hard to determine their genomic potential because we don't know what many of the genes are because there aren't knockout libraries. What's the process that you go through in choosing a new organism, right? If we move beyond Shigella and E. coli, what would be the next lowest hanging fruit? Maybe Danielle?
No, that's a tough question. I was still thinking about the prior one. Can I answer the prior question?
You can answer the prior question if you wish. Yes.
So there's one more thing that we haven't touched on, I think, that is really going to help us get at this predictability, and that's been the immense power. I think I said the words machine learning, but the ability to take in all of this data. So if we can find a place to store it, and then give all these really brilliant computer scientists the ability to access this data, I think that there's going to be a lot we can learn. We've apparently recently solved the protein folding problem with machine learning, right? So there might be a little bit to go.
We solve that every 5 years, don't we?
Yeah,
we do.
But it's gotten so much more accurate, and definitely there's been such progress over time. And I think we're at the cusp of doing the same thing for systems and getting to be able to predict what will happen if you perturb a network in a particular way, because we have all of this data and all these observations that an algorithm kind of takes in and says, oh, it's likely to do this. So to that end, there's actually a paper that I was going to bring up, and I cannot pronounce the name properly, probably, but from Gokale, who is focused on finding ways to develop mathematics, so more first principles approach to figuring out what happens when you change the context for an organism. And I think that's a beautiful example of what we can do right now with the mathematics we have, and where we're going is going to be bringing in these much higher throughput data pipelines to use all the observations that we need to create a repository for.
All right. I like that.
So back to your question about organisms. So I would say, low-hanging fruit, B. subtilis, is highly engineerable. But I want to figure, how do we motivate people? So like I said, in the old days, they always said, don't work on those other organisms because they're too hard, and you got discouraged from that. At least I did. But I suspect it's similar. And so how do we motivate people to really start to explore the wonderful numbers of amazing organisms that populate the earth, like things that are radio resistant and things that are can grow in geysers, and maybe things that can grow on the moon and things like that. And I think that's going to be the next big thing. And there's one small opening there. I go back to the reef. There was a great paper about, people traditionally, whatever it is that makes reef is been, highly unengineerable. And recently, I've forgotten the name of the person. They were able to get CRISPR to work in these microbes, and that's been true in some of these odd microbes. Is the ability to use CRISPR technology might start to open up the ability to do more engineering and genetics and not have to rely so heavily on, say, plasmid-based introduction. That's one possibility.
All right. Well, let me wrap things up and ask you one more question each. If you could solve a problem in the world with synthetic biology, and your microbiological techniques that are available to you weren't hindering that, what would that problem be? And can you describe how you would solve it? Danielle, you go first.
I was waiting for Pam to start talking about the reef and how she's going to save the reef.
I'm fixated on the reef, so that's going to be my answer. You can go for it.
No, it's okay. I was like, I want to fix the reefs now. But
let's if we all went and worked on it, so instead of all toiling away in our little labs, if we had this massive effort, maybe we could solve the reef,
Yes.
My best friend runs an aquarium, so
I like that, invested.
Sorry, Daniel.
Well, tell me then, Pam. Pam, tell me what about the reef is it that you want to solve, and then I'll come back to Danielle.
Make it grow again.
Okay, so you think you can use synthetic microbiology to make
the shade? By the way, that goes back to the shade paper about resilience. How do we build resilience into the poor reef? It's dying out because of all these insults it's having from global warming. So I guess writ large, the problem to solve is to make plants, microbes, whatever, resilient to global warming, right? That's the big problem.
So that's temperature and carbon dioxide concentration, two massively difficult things maybe to achieve.
You want to solve the problem that the Earth. So I'm really into trying to fix the Earth, and I think that there's two things: fix the Earth and go to space, right? So going to space isn't going to happen for me, but maybe making a small contribution to the Earth or the ocean. So for me, it's really looking at those questions around the environment and or sustainability. So engineering, the uber goal is, if you can, microbes can help, but you got to increase plant yield to feed the world. And so if you could double the efficiency of photosynthesis, however you did that, that would be huge. So that's a synthetic biology problem, and microbes contribute to that.
Well, yes, you could engineer the root microbiome to provide more nutrients. That would be
or cyanobacteria or algae. So people have shied away. Photosynthesis is hard, and I guess what I want to do is inspire people to work on hard problems.
I like that.
So mSystems. So how do you do that? So the problem, right, is, oh my God, how am I going to publish papers if I work on something hard and I don't figure anything out? So maybe mSystems could reward people. I'm just riffing here. Could reward people who work on hard problems, but only make a very small finding. Maybe we could be the place for those kinds of things also.
I like that. How difficult was it to get to this point? And we'll give you credit to an extra ladder.
If you're working on a really big problem and you make only a little bit of progress, maybe that's a thing.
I like that. All right, Danielle, what's your next Nobel Prize going to be on?
So I hadn't thought of the reefs until Pam started talking about it, so that's why that came up. But
now you're obsessed with the reefs.
Now I'm like, wait a minute. No, but what I had been thinking about for a while now is, what can we do to enhance the consortia that are in compost heaps? What can we do to help the world recycle? What can we do to help the problem, not to fix it? We have to fix it too. What's already done is got to be repaired. But what can we do to help draw it back and go back to a world where...
And by the way, water purification falls in that.
Exactly. How can we sort of self-remediate? How can we have each home have microbes that are carrying out these processes and go back to living, more in community with nature rather than kind of spoiling it and then fixing and then spoiling and fixing and going through these cycles that are not really productive for us. So my dream is to make microbial consortia someday that can degrade all our plastic and also maybe make the new precursor that generates heat for our house in some way that doesn't produce CO2.
Well, how about this one? This came up in a conversation I was having. Have a consortia that make a plastic that they can also eat. And so you have a self-contained system. we had this idea on a venture capital call, so somebody may actually do it. But you have a totally self-contained system for making and degrading the plastic.
Yes, exactly.
I like that. And
there you go.
And, on that note of inspired bioethical designing products which have a lifespan, I think we can wrap it up. We've covered virtually everything and been to the moon and back. So I think it's wonderful. Thank you very much, professors, and an opportunity to discuss synthetic microbiology in our first episode. I think that was wonderful. And remember, this episode is brought to you by mSystems, the journal, and it's available at msystems.asm.org. I'm your host, Jack Gilbert. Thank you very much.